Okay, so hello and welcome everyone to today's live class session. I will be discussing today the week four live. I'm taking the live session of week four contents on the course Experimental Biochemistry, which is being taken by Professor Sagata Dasgupta and Professor Shomo De of IIT Kharagpur. I am Srijan Sen, and today I will be taking your live session of week 4 and discussing the contents which was taught to you in week 4 and also solving a few assignments and questions which will be helping you in solving your assignments. Okay, so we will be starting with our today's lecture. Okay, so let us begin. So today we would be discussing mostly about proteins, after which we would be discussing the different concepts of the proteins, the structures which these proteins can form. We will be talking about the primary, the secondary, the tertiary. Then we will be speaking about the quaternary structures of these proteins and then how these proteins are being formed as to what are the interactions when these proteins are forming. So we will be discussing about this, after which we will be taking over with the denaturation of the proteins and the renaturation of a few proteins, and then we would be solving a few questions related to your assignments. Okay, so without delaying further, let us start. Um, so what are proteins? Proteins So proteins are biopolymers which are made up of 20 alpha amino acids linked by peptide bonds. So we have previously in our previous live sessions as well as in your previous course contents, you have already studied about the amino acids and about the different aspects of these amino acids. The different types of amino acids are present and about the characteristics. Some are acidic, some are basic, some are hydrophobic in nature, whereas some are hydrophilic, whereas some have aromatic ring carbons in their side chains, also some have aliphatic side chains. So proteins are biopolymers which are made up of these, and these all amino acids are 20 in number. So these proteins are a combination of these 20 alpha amino acids which are linked by peptide bonds. So these proteins are this protein backbone is actually a polypeptide backbone, which is a repeating sequence of NCC. So what are these? I will be coming to it. So it is actually N, which is the your amine group. After which you have your C alpha, then you have your carboxylic carbon. After which the thing repeats actually. So this is a repeating sequence of your polypeptide backbone. Now, the side chain or the R group is not the part of the backbone or the peptide bond. So, it says that the backbone structure is formed by this three carbons, three atoms for each amino acid. So, these three atoms repeat itself to form the backbone of your protein. Whereas your side chain is not involved in this backbone formation of your proteins. So, how is this polypeptide chain formed, and what is the peptide bond? How is the peptide bond forming? So, if I take two amino acids, say I take two random amino acids, and then you undergo the dehydration reaction. Okay. So what is this dehydration reaction? You are losing water. So how you are losing water is that, so this is say carbon with R group 1, and then this is a amino acid which is having a side chain with say R2. So now you are losing a water. So how this process takes place? So the OH is lost from the carboxylic acid, and an H is lost from this amine group, and then the dehydration reaction takes place, and followed by you are getting the peptide bond. Okay, so in this manner your peptide bond is formed. Okay, so in this manner the process actually repeats, and if you can see from this structure which I was telling in my previous slide about the backbone, that is NCC. So this is your alpha carbon. So this is your alpha carbon, and this is a part of your carboxylic acid. This is a part of your amine, and then the process gets repeated. This is another your alpha carbon. Okay. Now, if we take two or three different amino acids, and if we take two to three different amino acids, say we are taking methionine, then we are taking aspartic acid, then we are taking leucine, then tyrosine. So in each of these step, first the two amino acids come close to each other, and then they are losing your water and joining together by your peptide bond or the amide bond formation. Then another amino acid comes and joins via the loss of this water molecule and another molecule, and this process keeps on repeating, and then you get a series of peptide bonds and a series of amino acids joining together to give you the primary structure of a protein. Now, the polypeptide backbone, as I am speaking, is formed of your nitrogen coming from your amine or your N terminus. Then this is your alpha carbon to which your side chain is attached, followed by your carboxylic acid group carbon, and then the thing gets repeated in the similar manner. So it's NC alpha C, NC alpha C, and in this pattern you get the polypeptide backbone. And the sequence is written from the N terminus to the C terminus. So like here there are four amino acids forming your protein structure. So we have methionine, then aspartic acid, then leucine and tyrosine. So the sequence of this protein would be methionine, then aspartic acid, leucine and tyrosine. So it's written from the N terminus to the C terminus. Now what are the characteristics of this peptide bond? So these peptide bonds are covalent in nature, and we know that covalent bonds are strong bonds. These peptide bonds have a partial double bond character. That is, the lone pair of nitrogen falls back on the carbon, and then there is a partial double bond character between them, and also there is electron delocalization which occurs. So it has a distance. So how can we tell that? So it has been experimentally determined that the distance between these two bonds are around 1.32 angstrom, which is midway between. So we can tell it's the average between your single bond, which is usually 1.49 angstrom, and a double bond, which is 1.27 angstrom. These peptide bonds are rigid and thinner, so so the backbone of a protein is present on the same plane. So that's why we can call this bond to be planar and rigid. So it prevents your transconfiguration of the neighboring alpha carbon atoms, and the side chains are free to rotate on either side of the peptide bond. So these are a few of the characteristics of your peptide bond. So, from this slide, you can expect a few questions related to your peptide bond formation regarding tick the correct options which you feel is so you can expect a few questions like tick the correct options which you feel is correct for as the characteristics of the peptide bond. So then you are given a few options and then you have to tick the correct option. And maybe one of the option would be incorrect or can be an MSQ question where you have to take the correct answers. Okay, so the typical bonds in a protein molecule includes the covalent bonds among which we have the peptide bond and the disulfide bond. Then from the non-covalent bonds we have the hydrogen bond, the ionic bond and the hydrophobic interactions. So, so what are these disulfide bonds? So disulfide bonds are formed between two cysteines. So firstly when two cysteines comes close to one another, the so cysteines have a SH group at its in its side chain, and these two cysteines come in close proximity and undergo oxidation to form this disulfide bonds, followed by the loss of two hydrogen and two electrons. So we call the single molecule as cysteine with a TeIne, whereas the disulfide bonded cysteine is called CYSTINE. So this there is a difference between the two and in spelling as well. So these bonds are actually important in the folding of the protein as well as in giving different structure to the protein. So these bonds are actually important. So it is formed between two cysteine R groups of the polypeptide chain. So it is formed between the two SH groups. Now what we see on the right hand side is the structure of insulin where we have so basically insulin has two chains. So the pre-proinsulin has three chains which is A chain, B chain and the C chain. So what we see here is the amino acid sequence of your bovine insulin. So the two polypeptide chains are joined by your disulfide cross linkages. So the A chain is identical in humans, pigs, dogs, and rabbits, and your sperm whale insulins, and the B chain of the cow, pig, dog, goat, and horse are also identical. So what is important over here is the two chains are joined by the disulfide cross linkages. So therefore, you will be finding that in proteins and in the folding of proteins as well as in the structure determination of proteins, these bonds are very important, these disulfide bonds. Now coming to hydrogen bond, so nothing much to say about this bond because most of you are aware of it. So hydrogen bond is formed between a electronegative atom and the hydrogen. So hydrogen bond is formed between an electronegative atom and an hydrogen. So this electronegative atom can be your oxygen, can be your fluorine, can be your nitrogen, can be any of your electronegative atoms. Okay, so the hydrogen bond is also an important bond in your protein, in your proteins, and it helps in the determination of your structure as well, because it will be important when these side chains actually interact with the solvent molecules when these proteins are in your solution form. Okay, so now coming to the ionic bond formation. So if we can see from here that these ionic bonds are also important in some kinds of interaction between the two amino acids present at a certain location. So proteins are a collection of amino acids. So these ionic bonds via your electrostatic interaction comes close to one another and are important in determination of your protein structure. So so pH actually influences the strength of your amino acids. So depending on the pH, you can tell about the charge on a particular amino acid, which we have already seen in our previous lectures as to how pH affects the charge on the amino acids, and how the characteristics of the amino acids, how information of the about the characteristics of the amino acids are actually important, because the basic amino acids would be having a pi in the range of something around 8 to 10, whereas your acidic amino acids would be having a pi at around your 3 to 5. So these informations about the characteristics of the amino acids are important. So here we are seeing is glutamic acid and arginine, where arginine is a basic amino acid and your glutamine is a negatively charged and an acidic amino acid, and how these two amino acids or these types of amino acids are actually important for your ionic bond formation. And pH is an important factor in determining the strength of your ionic bonds. So coming to the next one, which is your hydrophobic interactions. So hydrophobic interactions are important in the formation of your protein structure, and so there are a few polar moieties as well as your non-polar moieties, and this is the unfolded polypeptide chain. And so the hydrophobic, what is this hydrophobic interaction, is that hydrophobic amino acids get themselves try to move away from your solvent and tries to form the core of your protein, keeping the polar amino acids or the polar side chains to exposing them to the outer environment, that is to your solvent environment. Okay, so these hydrophobic interactions or the hydrophobic amino acids or the side chains forms the core of the particular protein, and this is also an important interaction, rather a very important interaction in your protein folding as well as in your protein refolding and this type of thing. So these interactions are also a strong interaction and are an important interaction for your protein structure determination. Now coming to the peptide bonds in this summary, so we have heard about the hydrogen bond, where we see it is between the hydrogen which is attached to an electronegative group and an electronegative atom, which can be your oxygen, can be your fluorine, can be your nitrogen. Then we have seen about the ionic bonds, and we can even see from here that even when two amino acids are far away from each other, if there is an electrostatic interaction between them, or if two amino acids are oppositely charged, they can form these ionic bonds. Also, we have seen about the disulfide bridges or your disulfide bonds, which can be formed between the two cysteines if they are at close proximity to each other. And so this is also an important bond. And also we have seen about the hydrophobic interactions, and we have know that van der Waals interactions are some weak interactions which also takes a part in your protein structure determination. So why are we always speaking about this protein structure? Because uh, when a protein is in unfolded state, it is not in its active form. A protein can only be activated. We cannot say not only be activated, because there are a few polypeptides which are also few small polypeptides which have their actual function, but these are small molecules. So proteins we usually think of is a big molecule which is activated in its folded state, and these folded proteins actually have functions to play in their daily life. Now coming to the structure of the protein, so we have namely four different structures. So we can classify them as the primary structure, then we have the secondary structure, followed by a tertiary structure, and finally we can speak of the quaternary structure. So the native protein structure is essential for the biological function of a protein, and loss of structure results in the loss of your biological function. So what is a primary structure of a protein? It is the linear sequence of amino acids forming the backbone of a protein. So if you join a few amino acids in a linear format via your removal of your water molecule, and you try to say build a chain of amino acids, random amino acids, 20, we have 20 amino acids, so you can build up a random chain of 20 amino acids and in a linear format, so that we call as the primary structure, in which you would be having your N terminal. So the first amino acid would be called as the N terminal amino acid, and then the last one would be called your C terminal amino acid. Okay, so a linear sequence of amino acids joined by your peptide bonds is called as the primary structure of your protein. So peptides can be dipeptide, tripeptides, tetrapeptides. Then you can have oligopeptides, which can be up to your 20 amino acids. Then you can have your polypeptides, which can range between your 20 to 50 amino acids, and proteins, which so proteins have a varying range of molecular weight. So the molecular weight comes into the scenario when we have the varying ranges of amino acids as to the varying numbers of your amino acids. So proteins can be of different molecular weights and having different functions. So it can vary from a very vast. It has a very vast range. Can have 50 amino acids. Can have even 5,000 amino acids. Okay. So a single amino acid chain in this polypeptide chain may actually change or completely abolish your protein function. So one specific example of this is that when you're in the normal red blood cell, we have this sequence of DNA, and just a single change in your codon, or a single change in the amino acid can actually affect your can actually lead to a disease condition. So here we see a normal beta globulin, or or a normal blood cell, where we have the amino acid sequence to be a threonine, proline, then we have the glutamic acid followed by another glutamic acid. Okay, so but the mutant one, when this glutamic acid changes to your valine, when this glutamic acid changes to your valine, this normal red blood cell becomes a sickle red blood cell. And this condition, we have heard about it as the sickle cell anemia. So this is a condition where a change, a minute mutation in one amino acid is actually disrupting your body function. Now, nextly coming to the secondary structure. So, secondary structure is the regular arrangements in space of adjacent amino acid residues in a polypeptide chain. So, there are three types of secondary structures, namely your alpha helix, then you have your beta sheets, and then your beta turns. So what is an alpha helix? So it is formed as a result of the hydrogen bonds between your carbonyl carbon and the amide hydrogen of the polypeptide chain. and it does not depend on the side radical. So it is the interaction between the carboxyl oxygen and the amide hydrogen. So how far can these amino acids be present such that they can actually form a alpha helix? It is that they have to be they can be present at a distance of 4 amino acids. Like if we are considering an ith amino acid and an I plus fourth amino acid, so the ith carbonyl oxygen can interact with the I plus fourth amino your amine hydrogen in forming and will enable the forming of your alpha helix. So the carbonyl carbon of each peptide bond is linked to the hydrogen bond with the amide hydrogen of the fourth amino acid towards the C terminus. Okay, so in this manner, your alpha helix is being formed, and your side radicals are have the ability to rotate about this peptide backbone. So they are not actually this does not affect your alpha helix formation. So this alpha helix, okay, and another thing is that the two, so these alpha helix have a 3.6 residue per turn, and they have a pitch of 1.5 angstrom. So the features of the alpha helix includes they have a most common and stable conformation. They have a spiral structure, which you can see from this. That also you can see from this that they have a spiral structure, and the pitch of this is three. It has a 3.6 residues per turn, and the distance between the two residues is 1.5 angstrom. So the total distance between these two turns is actually 3.6 into 1.5, which is a 5.4 angstrom. Okay, so now the spiral, it has a spiral structure, that is the polypeptide bonds from the backbone of the spiral and forms the R groups of the amino acids remains outward of this spiral backbone, and these alpha helices are stabilized by your hydrogen bonds, and hydrogen bonds are weak bonds, but they determine the structure of your alpha helix. Now, how this alpha helix, or what are the problems which these alpha helix can helices can face, or which amino acids are not suitable in the formation of your amino acids, or how can these alpha helices gets disrupted with the presence of different amino acids? Okay. Now, if we see that the presence of proline, if we can get back and recollect the structure of a proline, it has an imino group, and it has a very what you call has a very folded structure, and not a very folded structure, it has a very rigid structure. That's why it does not have that flexibility, it has the least flexibility, and that is why the presence of proline is actually disrupting your formation of the alpha helix. Also, overcrowding of equally charged amino acids, especially your say lysine, arginine, histidine, or your glutamic acid, aspartic acid. So similarly charged radicals present or overcrowding of these similarly charged amino acids can affect your alpha helix formation. Also, crowding of bulk R groups like your leucine, isoleucine, the tyrosine can actually interfere with your alpha helix formation. Although these side chains are present outwards, but if we consider the alpha helix formation on a whole, these long chains or your bulky R groups would be affecting in a different manner and disrupting your alpha helix formation. Then your prevailing of amino acids with R groups which are capable of forming hydrogen bonds, and the presence of chemicals tending to form hydrogen bonds would also be affecting your alpha helix formation. Now coming to the next type of your secondary structure, which is your beta pleated sheets. So there are two types of beta pleated sheets. There we have the anti-parallel beta sheets and the parallel beta sheets. So the parallel beta sheets run in the same direction with longer looping sections between them, whereas the antiparallel beta sheets have the polypeptide chains running in the opposite direction. So both of these models are equally present in proteins, but the antiparallel structure is more stable compared to your parallel beta sheet. Now the question is why? But if we see from the structure, is that the alignment. First thing is the alignment, like the you say the NH, which is actually forming the hydrogen bond with your um, CO, that is the oxygen of your carboxylic group. So these are aligned very in a very proper manner. The alignment is so good that so this alignment is actually determining the strength of the bond. So this is actually adding to the stability of your anti-parallel beta sheets. Whereas if we see from the parallel beta sheet point of view, is that they have longer looping sections, and the distance between the hydrogen and the oxygen is somewhat in a very, you can say, a crooked manner, or the distance between them increases thereby. So as the bond distance is increasing, although we know it's a hydrogen bond, but a few or millions of hydrogen bonds can actually lead to strong bonds. So these minute bond difference is affecting the stability of your beta sheets. So therefore we can tell that antiparallel beta sheets have higher stability compared to your parallel beta sheets. Now coming to the beta turns, so beta turns, it permits the change of direction of the peptide chain to a folded structure. So beta turn or loops allows for the protein compaction, since the hydrophobic amino acids tend to be in the interior of the protein, where hydrophilic residues interact with the aqueous environment. So like we have two beta sheets, and then it is connected by a beta turn. So two beta sheets cannot be connected in any manner. So it has to be connected by some kind of turn such that they run anti-parallel to each other. So this is actually helped by your beta turn. And also this beta turn is necessary because this also helps in what manner would the amino acids position themselves. The hydrophobic amino acids would like to be in the interior of the protein, whereas the hydrophilic residues would want to stay outwards to the aqueous environment. Now coming to the next one, which is the tertiary structure. So it gives a complete idea of the three-dimensional shape of the protein, and it is important for determining the conformation of the protein. So it represents a spatial relationship of the different secondary structures to one another within a polypeptide chain, and how these secondary structures arrange themselves in the three-dimensional form of the protein. That is also important. That also this tertiary structure gives us an important idea about these things as to how the secondary structures are arranged inside a protein in the three-dimensional environment. And the spiral regions represents the sections of the polypeptide chain that have alpha helical structure, while the broad arrows represents the beta pleated sheet structure. So these yellow arrows are your beta pleated sheets, whereas the brown or the maroon spiral structures are your alpha helices, and these are connected by your loops, or you can call your turns. So this will tell about, and these tertiary structures are also important and vital for these three-dimensional protein structure is important for the functioning of your body. So the tertiary structure describes the relationship of different domains to one another within a protein. So firstly, if we see that the first we have a primary structure, then these primary structures via the different bond formations, your hydrogen bonds is forming a secondary structure. Now a few secondary structures having a relationship between them forms the domain, and then it gives you a tertiary structure. Now, a domain is the basic structural unit within a protein molecule. It is a part of the protein that can fold into a stable structure independently. And this protein domain can possess different functions. These proteins can have one to many domains depending on the protein size. A polypeptide with 200 amino acids consists of two or more domains, so that is variant as to how many domains you would be having. The most important thing is that it is the basic structure within a molecule which has an ability to fold within itself independently and then carry out different functions, and these domains are connected via various, you can call the flexible regions of the proteins. So this is a picture of a pyruvate kinase, a monomeric protein which is having your three domains which are given three different colors. So protein can have one, two, or as many number of domains it can have, connected via a few flexible regions, via a few many interactions, and then giving you a total tertiary structure of the protein. Now, the tertiary structure is based on the different types of interaction between the side chains of the peptide chain. So, what are these interactions? Includes your hydrophilic interaction to the water. Then you have the hydrophobic interaction, which occurs at the core of the protein molecule. Then we have the disulfide bonds, followed by your hydrogen bond, the salt bridges, and then obviously we have weak interactions like your van der Waals interaction, etc. So these or these various interactions actually determine the structure of your structure and functioning of your protein, and also these interactions would be important in the folding, refolding of your proteins, and how these interactions can be disrupted to get a linear structure of a protein, or your, you can call the denaturation of a protein. We will be coming to these things in the future slides. Moving on to the next one is the stabilizing interactions of your tertiary structures. So we have the hydrophobic interactions, which is the attraction between your nonpolar alkyl and your aromatic groups, forming a nonpolar center that is repelled by your water. So then we have your hydrophilic interactions, which is the interaction or your attraction between the polar or ionized R group and the water on the surface of the tertiary structure. Then we have the salt bridges, followed by your hydrogen bonds, which occurs between the polar side groups of the amino acids, and then we have the disulfide bonds, which occurs between your two cysteine amino acids. So these are the few stabilizing interaction of your tertiary structures, and here you can also expect a few questions related to it. You would be given a few interactions, and you would be asked to comment on the which of the importance of the interactions in the formation of the various structures. So this slide would be important for you. Now coming to the quaternary structure. So quaternary structure, it is built of a single polypeptide chain, the monomeric proteins, and your oligomeric proteins are built of more than one polypeptide chain, which you call as the subunits or monomers. Now, these quaternary structures describe the joining of two or more polypeptide subunits. Now, these subunits have their own tertiary structures which are joined together to form the quaternary structure, and there occurs the bonds, that is your non-covalent interactions. Then these subunits can either function independently, or for example you have a few say two to three monomers which join together, these two to three subunits which joins together to give you a quaternary structure. Now it can happen that each of these subunits have their function independently and can work independently to have different functions. Also, it might occur that only when these subunits are present cooperatively, or when these three subunits join together, only then this protein can actively function. So, and when one of these subunits is lost, it can actually result in the loss of function. So these can occur. So they can work independent. Subunits can work independently as well as they can work cooperatively. One example of these quaternary structure is your hemoglobin. So it is a globular protein which consists of your four subunits, including your two alpha and two beta of two different types of alphas and betas. Now, each subunit contains a heme group for your oxygen binding, and this binding of oxygen to one heme group actually is facilitating the binding of the other oxygens to binding of oxygen to the other subunits. So, just as I was speaking, so one subunit can work independently, and this can even activate the functioning of your other subunits as well. So this is one example where when the oxygen is binding to one heme group, it is facilitating or enhancing the oxygen binding to the other subunits as well. And then the replacement of even one amino acid in the primary structure with another amino acid is critical for the function of the protein. We have seen that previously about your sickle cell anemia, where glutamic acid is being mutated to valine, and we are seeing a diseased condition. Now, here we comment that protein structure is hierarchical. That is, first we have a primary structure which is important to give you the information about the sequence of the proteins, as to which of the amino acids is present in the N terminus, which amino acid will be forming your alpha helix, which amino acids will be forming your beta sheets, and then how would these and which amino acid is forming your C terminus. So you can comment on the sequence of the protein. Then we have the secondary structure which is associated with your local folding and giving you structures like your alpha helix, then your beta sheet, then you have a few turns and loops. After which we have the tertiary structure, which is your long range folding effects, like two or three secondary structures is forming a domain, and these few domains are interacting with each other via the different bonds and few other interactions, giving you the tertiary structure. And these tertiary structures can function even independently, or can work or if they have to work collaboratively, then they form quaternary structures. That is, they are important for your multimeric organization, and these quaternary structures can even go further and further for giving you supramolecular structures as well. Okay. Now, structure determines function. So these, now we see that these quaternary structures, when they go on to form supramolecular structure, or even in the tertiary structure or the quaternary structure, because we have seen that a few domains can work independently. So these tertiary structures form the quaternary. Quaternary forms your supramolecular. So each domain or your each structure has a particular function. So what the functions could be, so it can be your regulation or your some signaling, cell signaling, some regulations where you have to activate a particular kinase, or you are undergoing a few post-translational modifications. Okay, then other functions is includes your transport, then the structure determination followed by your movement, then catalysis. There are different functions. So structure of the protein is important for determining the function. But we have seen come across a problem which is known as the protein folding problem. So what determines the structure? So the two factors which determines your structure is your energy and the kinematics. So how we determine the structure? So there are different ways of structure determination. There can be your experimental methods where you undergo a few cleavages to determine the particular amino acid at the N terminus, and in this pattern you start cleaving the N terminal amino acids and find out the structure of the protein. Also you can do this with your C terminus, like there are enzymes or there are enzymes which actually cleave at different sites, and then you can comment on the structure of the protein. You get to know the sequence. Also there are computational methods. Then you have which will also help you in determining the structure. Then you have extra crystallography, NMR spectroscopy, etc. So there are different ways of determining the structure of your protein. So the protein folding problem states that the amino acid sequence of a protein determines the 3D fold, and this was said by Affinson. So some exceptions which includes are that all proteins can be denatured. Some proteins have your multiple conformations, and some proteins get folding help from your chaperone. So the function of a protein is determined by its 3D fold. So can we predict the 3D fold of a protein given its when the amino acid sequence is known to us? So if we know the amino acid sequence, and if we know that what sort of interactions can occur between the amino acids, what type of amino acids are present. So primary idea of amino acid, the primary idea or the primary sequence, the basic information about the primary structure of the protein can actually help you in building up the 3D structure of your protein. May not be it is accurate because we are building it in our human mind, but if we do it computational, there are computational softwares which actually develop the 3D structures from these primary structures, which will also help you in commenting on your the helice, your folding of protein as well. Now coming to the Leventhal paradox. So for example, we have been given a small protein, say of 100 amino acids, and we assume that there are three possible conformations of or the peptide bond formation. So if we see that in how many ways or in how many conformations of this can occur, so it is 3 to the power of 100, that is it is taking about 5 into 10 to the power 47 conformations. Okay, so even if we consider that the fastest motion is 10 to 15 seconds, so it would take about 5 into 10 to the power 32 seconds, which is around 1.6 to 10 to the power 25 years. But the thing is that if this is the truth, then it would be taking years to only comment on the structure of the protein. But we see that every year we are actually getting new proteins, developing new proteins, commenting on their structures, getting their sequence, and so what is the actual myth of this? So each protein folds quickly into a single stable native conformation, which is said by the Leventhal paradox. So there are only some stable conformations which these proteins can take, and this actually reduces the number of conformations, and therefore it also reduces the time factor as well. So therefore this is the concept of Leventhal paradox. Now we can comment on the energy as well. So there are different types of non-covalent interactions among the biomolecules, like your hydrogen bonds between the neutral groups, then between the peptide bonds, which have these hydrogen bonds have a strength of 3 to 7 kilocalories per mole. Then we have your ionic interactions, which is your accounting for 10 kilocalories per mole. Then we have hydrophobic interactions, which accounts for 1 to 2 kilocalories per mole. Then we have van der Waals interactions of about 1 kilocalorie per mole, and disulfide bridges, which accounts for 51 kilocalories per mole. So there are different interactions, and we can comment on the strength of the bonds or these interactions depending on the strength data which is given to us, and the different interactions which can occur between the two proteins. Now, if we have only your ionic interactions or only a few interactions with those two proteins becomes less stable. Whereas if we have a few series of interactions, say your ionic bond interactions, then your hydrogen bonds, then your hydrophobic and van der Waals interactions. So as many number of interactions occurs between the two proteins, it also increases the strength of your structure, or the two moieties actually becomes much more stable and a stable complex is being formed. So these are experimentally determined hydrophobicity levels. So if we see that which of these are more hydrophobic or your less hydrophobic, so higher the number of, so higher the number in the positive direction, so higher is your hydrophobicity. So we see that tryptophan, then isoleucine, phenylalanine are hydrophobic, highly hydrophobic. So these would be important in forming the core of the protein. And then we have your leucines, cysteines, methionines, valine, then your tyrosine. So then the hydrophobicity level decreases and we go to a negative value where these are more of hydrophilic in nature than your hydrophobic, and we see that arginine is the most hydrophilic residue and your tryptophan is the most hydrophobic residue from the given data. So this data was published in the year of 1983 by Forscher and Pilska in your European Journal of Medical Chemistry. And so these are some of the determined data which you may not keep in mind, but it is actually important because say in folding every amino acid participates. So as we were trying to find out that if from the structure we can comment on the, from the primary structure, if we can comment on the 3D structure of the protein. So these amino acid combinations or a knowledge about the hydrophobic levels of these amino acids can actually help you in a building 3D structure in your uh, pen and copy, or you can build up in your mind as well. So okay, we can take a 2 minutes of break, after which we can start with the next slide. Okay, so let us restart. So now coming to the next topic is your denaturation of proteins. So if we see that the conformation of a native protein is marginally stable, because after all these interactions are strong, but sometimes these interactions are weak as well. So these interactions depends on your pH, then the solvent in which your proteins are. So we cannot call an interaction to be very stable in a in a biased manner. So the strength and weakness of the bond depends on the situation. So the conformation of the native protein is marginally stable, and the conditions differ from those in a cell, those from in a cell, and you can have varying conditions. Therefore they can cause your protein structural changes, resulting in the loss of three-dimensional structure needed for your function. So this is what we can call as denaturation, because when there is the loss of the three-dimensional structure, which is actually important for the functioning of the protein, when these structure is lost, then this the denaturation of the protein is what we call it as. Now in the denatured state, the conformation of the protein need not be completely randomized, and a number of physical and chemical agents can cause this protein denaturation. A classic agent is heat. So one of these factor or for denaturation is your heat, which has complex effects on many weak interactions in a protein, primarily these hydrogen bonds. So the heat actually breaks these hydrogen bonds. So now on heating, a protein's conformation generally remains intact unless an abrupt loss of structure occurs over a relatively narrow temperature range, which we call as the TM. So above this TM, you can see an abrupt loss in the structure of the protein. So the temperature in which your protein is half natured and half denatured, which is what we call as the melting temperature. Okay, that is called the TM. So we can see that for ribonuclease, if we can draw a line on this, then we see that the TM of ribonuclease is somewhat around, you say, 58 degrees centigrade, whereas for apomyoglobin the TM is around, you say, 66 or 67, somewhat this. Okay, so this will so this TM actually determines as to how the structure would be lost, as to how far the structure would be lost when we are heating it. So on heating, the structure gets lost because it breaks the hydrogen bonds. Now this abruptness in the loss of the structure tells us or gives us information about that it is a cooperative process in which the loss of structure in one or more parts of the protein rapidly destabilizes the other structure of the other parts as well. So these abruptness about the loss in structure tells us about that this is a cooperative process in which loss in the structure of the protein will completely destabilize the other parts, and ultimately it will denature or destabilize the other interactions as well, and will completely destabilize the protein, leading to non-functionality of the protein. Okay, now proteins can also be denatured by extremes in pH. So this is another factor where your pH is playing a role. Then you have other miscible organic solvents, including your alcohol and your acetone. Okay, then you have agents like your urea, then guanidinium hydrochloride, which are actually you're reducing agents, and it reduces the different bonds, especially you can tell your disulfide bonds and other bonds which were formed by oxidation, so it reduces a few bonds. So these are another agents which actually denatures your protein, and there are other detergents as well. Okay, so these are a few of the things which actually plays a role in the denaturation of your protein. So none of these agent breaks your covalent bonds. So this is an important thing, that none of these denaturing agents can actually break your covalent bonds. So organic solvents like in urea and detergents act primarily by disrupting your hydrophobic interactions that produce the stable core of your globular protein. So it disrupts the hydrophobic interactions. Also, urea and guanidinium hydrochloride disrupts your hydrogen bonds. Then pH can alter the net charge on the protein, causing electrostatic repulsion and the disruption of your some of the hydrogen bondings. So how each of these factors are actually affecting or denaturing in your protein, it is given over here. Now these denatured structures resulting from the various treatments may not be the same. Lastly, these denaturation results. So how do we know that the protein has denatured completely? It is occurs because it results, this denaturation results in aggregation and precipitation of the unfolded protein, because the hydrophobic residues are exposed to the solvent, and these hydrophobic amino acids do not like the water molecules, so they will ultimately aggregate and precipitate down. So from this we can tell that the proteins have denatured completely. Okay. Now coming to the renaturation of protein. So there has been previous studies examining the refolding of an enzyme called ribonuclease, which provided an experimental proof that the tertiary structure of the protein. So a study on the refolding of ribonuclease showed that the tertiary structure of your protein is actually determining your primary structure. So what was done in this experiment is that a purified ribonuclease was firstly denatured to its unfolded or your inactive state. So this is the structure of your say ribonuclease, and then by the addition of urea or beta marcaptoethanol, so BME, these are the reducing agents which so this was a state which was catalytically active, and now on the addition of becomes an unfolded state and an inactive state, so where the disulfide cross linkages are reduced to your cysteine residue. So the denaturation to its unfolded state was done by its exposure to a concentrated solution of urea and reducing agents like beta-mercaptoethanol. Now what happened? This unfolded protein and the reduced disulfide cross links when have been, now the protein is in the unfolded state now. Now when these conditions are removed and the unfolded protein is brought back to from a state where we do not have urea and your mercaptoethanol, so the randomly coiled or the denatured ribonuclease spontaneously refolded to its catalytically active form. So when the conditions or chemicals were removed from these unfolded state of the protein, from the proximity of the unfolded state of the protein refolded itself to its native form, which was the active form, via your correctly reformed disulfide cross linkages. Okay, now even the protein's four unique disulfide bonds reformed correctly. So this experiment provided the first evidence that the amino acid sequence of the polypeptide chain contains all the information required to fold the chain into its native three-dimensional structure. So there are So if there are eight disulfide or there are eight SH or there are eight cysteines, so they are capable of forming four disulfide bonds. So there can be different combinations of disulfide bond formation. But we see when the protein A was removed from the proximity or removed from a condition of urea and reducing agent, it reformed itself, forming correct disulfide bonds which was previously present in the native structure. So therefore, this was the experiment which told us or gave us information that the primary structure of the amino acid is important in determining your three-dimensional fold of the protein. Now coming finally to the thermodynamics of the protein folding. So commenting on the thermodynamically, thermodynamically is the protein folding process can be viewed as a kind of free energy funnel. So the unfolded states are present on the top of the funnel and are characterized by a high degree of conformational entropy and relatively high free energy. So when the in an unfolded state the proteins have higher entropy and have the ability to move here and there, they are highly flexible. So higher the flexibility, higher will be your conformational entropy, and therefore but as the folding proceeds the narrowing of the funnel reflect on the decrease the conformational space that must be searched as the protein approaches its native state. So n is the at the bottom of the funnel, so small depressions, you can see a few small depressions along the sides of the funnel. So these are the semi-stable intermediates which can form because of the folding process. So then again it unfolds and again refolds itself. So there are different intermediate states among these total from a unfolded to a folded state. You will be finding a few intermediates where the protein folds and then again it denatures because it finds itself in a very uncomfortable position and then it again refolds to form a the folding process and then finally the folded protein is formed. So the wider or the broader region at the top your will be of your unfolded proteins, and as the folding process continues the funnel gets narrower. Now these funnels can have a variety of shapes depending on the complexity of the folding pathway. So these funnel tells or gives us information about the folding pathway, then number of semi-stable intermediates present, and then the potential for particular intermediates to assemble into aggregates of the misfolded protein. So a few informations from this funnel can be gathered, and also the thermodynamics of the protein folding can be understood from this funnel model or this inverted funnel system. Okay, so with this we would be ending our lecture. I would be discussing a few questions with you people. So how many people do we have? So if you I see only one. If you are present, Aarti, can you please respond or text me that you are present over there? Okay, that's nice, good. So what I would be doing is that just we only have one participant. So maybe you can answer I will be showing you the questions and then you can answer them in the text only, and then we would be discussing. So this would be a bit. This is being recorded for other students as well who are not present, such that, and it will be uploaded in the YouTube as well. So we would be discussing in that manner. So you can take some time in answering the questions. Okay. So coming to the first question, it says the beginning of the peptide chain has the beginning of the peptide chain has the alpha carbon, carboxyl group, amine group, or the carbonyl group. Yes. So you say it's C. So that is correct. So the beginning of the protein starts with the amino terminus, that it has the amine group at its N terminus, and the end of the protein is the C terminal C terminus. So it has the carboxylic acid group. So yes, C is the right option. Okay. Now the next question says, a polypeptide chain is generally referred to as the. So whether it comments on the primary structure of the protein or the secondary structure of the protein or the tertiary structure or the quaternary structure. What is a, it is a polypeptide chain, and what is it referred to as? This one, this is the correct option. Okay. So you say C, but if you look back to the slide, the primary structure actually constitutes the polypeptide chain, which folds in a specific manner to give you the secondary structure, and these secondary structures arrange among themselves or interact to form particular domains. Now these domains are actually important for your tertiary structure, but if I am speaking of a polypeptide chain, it is only a chain, it's the primary structure of the protein which is being referred to as. Okay, but it's okay, you tried, it's good. So, but the right answer is the polypeptide chain is referred to as the primary structure of the protein. Okay, the next question says, a peptide bond is from the elimination reaction of carboxylic group, condensation reaction between amine and carboxylic group, substitution reaction of the amine group by amide ligand, or the addition of H2O to the amine group. This is a easy question. I guess you can answer it right. Yes. So condensation reaction occurs between the amine group of one amino acid with the carboxylic group of the other amino acid. Yes. So condensation reaction, that is the loss of water is occurring. So that is the right answer. Okay. Now moving to the next set of questions. So the number of possible tripeptide sequence which can be formed are 400, 800, 4000, or 8000. So first we need to think that there are 20 amino acids present. So what are the possible or how many tripeptide sequence can be formed from these 20 amino acids? We have 20 L alpha amino acids and 20 are known to us. So how many tripeptide sequences can be formed? Obviously repetition of amino acids are allowed over here. So what would be that number be? This is a sum of your permutation combination. So we have three places or three chairs where 20 people can sit. So 20 people can sit in three places at the same time if we think, which is not possible, but if we think, so each place will have 20 possible combinations. So it would be your 20 for the first place, and then it was a dipeptide, then 20 into 20, that is your 400, and now comes another 20 for a tripeptide, so it is 20 into your 20 into 20. So there are 8,000 possibilities for your tripeptide sequence formation. Okay, so it's 20 into 20 into 20 for a tripeptide. So number of possibilities are. Okay, now next question says in the peptide bond the CN bond has. So what is so this question is on the characteristics of your CN bond or the peptide bond. So I was speaking in my slides about the characteristics of the peptide bond, and then you could expect a few questions. So what are the options? Only double bond character, only single bond character, both single and double bond character, and delocalized electron system. So which is the correct option, or which are the correct options? So what happens? Okay, single bond character. Okay, is it so? Okay, let's get back to the structure of where we were speaking about the. So where was this? Yeah. So if we see about the characteristics of the peptide bond. So these are covalent bonds and these are strong bonds. Also, we have a partial double bond character between them because the lone pair of the nitrogen will fall back on the carbon, giving this oxygen a negative charge. And there is also an electron delocalization which is occurring between them because there is a positive negative charge occurring, and there is a double bond delocalization which is also taking place. So this CN bond has the capability to stay in the single bond as well as in the double bond, and this has been also experimentally shown that the distance between this C and N is somewhat between your single bond distance and your double bond distance. So we cannot tell that it is totally a single bond or totally a double bond. So it has the nature of both. So it has a partial double bond character, and also an electron delocalization is taking place. Okay, so here the correct option would be both your C and your D. So both would be correct because it has a both single bond and double bond character, as well as your delocalization of the electron system is also taking place. Okay. The next question is disulfide bond is formed between. This is an easy question. I think you can answer that. So the disulfide bond is formed between two cysteine residues. Cysteine are serine residues, two serine residues, or the serine and threonine residue. Yes, so only cysteine has an SH group at its side chain, which oxidizes. So the two cysteines oxidizes to form this disulfide bond. So the two cysteine residues are forming this disulfide bond. So A is the correct option. Okay. Moving to the next set of questions. So the phi and the chi angles are formed around. Although I haven't spoken about the phi and the chi angles in this, but let's check if you can answer that. So it is the NHC alpha and the C alpha CO. We have the NHCO and the C alpha CO, or we have the C alpha NH and the CO C alpha and the NHC alpha and the NHCO. So what is it? Check if we have a structure of something. So this phi and the psi angles are actually because of your. Okay, so we have a structure over here. I can use this structure to explain you people. I don't have any other structure, so I would be. Do we have any other structure? So this is a good structure to explain. Okay, so see, so if we see, we are only looking at the backbone chain. Okay, only at the backbone atoms present. So in the backbone atoms of a protein or a peptide, what do we have? We have a nitrogen, we have a C alpha, we have a carboxylic carbon, then again we have a amino nitrogen, then we have a C alpha and then the carboxylic carbon, and this thing gets repeated one after the other. So if we see that or if we look that the okay. So these phi and these psi angles are called the dihedral angles. These are actually formed between four atoms. So four atoms are responsible for this angle formation. But in the options you will be seeing two, because between which two atoms is this or this angle present. So if we see that first we have is the CO and the nitrogen. So then, so how many angles can be there? So we have one over here. So dihedral angles are determined in this manner. Okay. Then we have between N and C alpha. So we have over here. Then we have between C alpha and your carboxylic carbon. So we have another angle over here. And then we have the same thing, which is your C carboxylic carbon and your amide nitrogen. Yeah, amino nitrogen. Okay, so we have basically these three. Okay, so the first one, that is between your CO, the carbon, and the nitrogen one, the carboxylic carbon and your amide nitrogen. So this is known as your omega. Okay, the next one, which is present between your nitrogen and your C alpha. Between your nitrogen and C alpha is your phi, and the angle between your C alpha and your carboxylic carbon is your psi. Just as I was speaking, these angles are dihedral angles and are formed between four atoms. So there will be, so these two atoms are the middle atoms. Okay, the other two atoms would be this one and this one for your phi, this one and this one for your psi, and for omega, for your because this angle is also your omega. So for this it would be this one and your this one. So now in the options you can get two atoms or maybe four atoms. So depending on that, you have to answer them. So don't get confused like why four atoms are given, although you have been taught about only one, only about the two atoms and the angle is present between the two atoms. Just because these are dihedral angles, so four atoms are actually involved in this angle formation. So the phi angle, another time I am repeating, so the phi angle is formed between your carboxylic carbon, the amide nitrogen, the C alpha and the carboxylic carbon of the ith amino acid. If this is your I minus 1th amino acid, this is your ith amino acid. Okay, and then your psi would be formed between your in between your nitrogen of your ith amino acid, then your C alpha, then the carboxylic carbon, and the Ni plus 1th amino acid. And for the omega, it is between your carboxylic carbon of your ith amino acid, the nitrogen of your I plus 1th amino acid, then the C alpha of your I plus 1th amino acid, and on this side we have the C alpha of your ith amino acid. So these are the four atoms which are involved in the forming of the angles of omega, then phi and psi. Okay, so now I think now it would be easy for you to answer the question. So the phi and the psi angles are formed around. What would be the right answer? So the right answer would be A. That is between your nitrogen of your ith amino acid and the C alpha, and the C alpha of your ith amino acid and the carboxylic carbon of your ith amino acid. Okay, so the right answer is your A. Although you can be given like two atoms or four atoms. So don't get confused by that. Okay, the next question says an alpha helix is stabilized by hydrogen bonding which is formed between the what? CO and NH of the ith and I plus 1th residue, C alpha and NH of the ith and the I plus 4th residue, CO and NH of the ith and I plus 12th residue respectively, and CO and NH of the ith and I plus 4th residue respectively. So what is the correct answer? Between which two residues can an alpha helix be formed? Or between which two residues can a hydrogen bonding result in the formation of an alpha helix? An helix is a spiral, an alpha helix has a spiral structure. I'm giving you a hint, it has a spiral structure. So the neighboring atoms would be in a spiral position, not favorable for hydrogen bonding formation. Rather, an atoms at far away distance would be in a better condition with forming a hydrogen bond. So this was also one of the characteristics of your alpha helix, that first of all the alpha helix is formed, or the hydrogen bonding is formed between your carboxylic oxygen and the hydrogen of your amino group. So that is correct. So C alpha and N, so B is in any ways eliminated from the race. Now the other options say CO and NH of your ith and I plus 1, ith and I plus 2, and I and I plus 4. So if you look back to my lecture and see, so in that slide I have told that it is formed between the ith and your I plus 4th residue respectively. These alpha helices have a 3.6 residues per turn, and each amino acid is at a distance of 1.5 angstrom, thereby resulting in the pitch of, or the difference between the two turns to be 3.6 into 1.5, that is 5.4 angstrom. If you look back to my slides, you can see it, and therefore D is the right option. That is, it is formed between the CO and NH of the ith and the I plus 4 residue respectively. Now, the next question is a protein has the following amino acids in its sequence: methionine, cysteine, glutamine, arginine, serine, proline, tyrosine, lysine, and glycine. So, which of the following alpha helix or helices are expected to be formed within the above sequence? So those who have come late or haven't heard my lecture, I'm just repeating the characteristics such that it is easier for you to attend answer the question. So proline is actually a helix breaker and is not favorable as an amino acid in the formation of your um, alpha helices. So when proline is present, it will actually disrupt your alpha helix formation. That is one thing, and then the next thing is that just because an alpha helix is formed, the hydrogen bond in the alpha helix is formed between an I and I plus fourth amino acid. Therefore, there has to be a minimum of four amino acids. Okay, so these are the two hints I'm giving you. Now, if you can find out the right answer. First of all, there has to be a minimum of four amino acids, and proline is not a good choice in the helix formation. Yes, A is the correct option because in any case C gets eliminated, and in B and D both proline are present, so proline acts as the helix breaker. So A is the correct answer. Moving on to the next question. When a polypeptide folds in an aqueous environment, the loss in entropy is compensated by hydrogen bond formation between water and polar residues, release of free water molecules, exposure of polar amino acids on the surface of the protein, release of proton from the acidic amino acids. So when a polypeptide folds, what actually happens? That is what we have to think. So when a polypeptide is folding, in the meantime, you can answer. I'm just giving you the concept of the protein folding once more. So when a protein actually or a polypeptide is actually folding, the hydrophobic moieties forms the core of the protein, whereas the hydrophilic amino acids forms the outer layer. So when the water molecules would be present, the water molecules which were actually present in the core of the protein, or the which were actually surrounding the protein, um, and the hydrophobic residues are forming the core, the water molecules have to be removed out. Okay, so from such that the hydrophobic molecules can make some space and form the core of the protein. So this is how the protein folding is forming. So what is the correct option? So the loss in entropy is compensated by, because when a protein folds there is some kind of loss of entropy which is not a favorable thing. So what is the phenomenon that is compensating this loss in entropy? Okay, so it is the release of free water molecules which were primarily present in the core of the protein. Say we are assuming the core of the protein to be vacant at first, although it is a it's a polypeptide, it is not in a folded state, but we are thinking that there is some water molecules, the free water molecules would be removed and the hydrophobic molecules would come and interact forming the core of the protein. So the release of water molecules is actually compensating the loss in entropy. Okay. The protein denaturation by heat and urea is mainly caused due to rupture of hydrogen bonds, rupture of hydrogen bonds and destabilization of hydrophobic interaction, displacement of the water molecules around the protein, or all of these. So how is heat and urea playing a role? Yes, all of these options are correct, because heat is also rupturing your hydrogen bond formation, then it is is also destabilizing. This urea is destabilizing your hydrophobic interaction. Also, these when these hydrophobic molecules or these hydrophobic amino acids are out in the environment um, or in the space where it can encounter the water molecules, it is also displacing the water molecules around the protein, and thereby it is leading to the aggregation and precipitation of your protein. So D is the right option. Okay. The next question says glacial acetic acid is added to two different proteins. Protein X of Pi9 and protein Y of Pi4 in equal amounts. Initially, both the proteins are at pH 7. Which of the following observation is expected to be observed? So those who have not been present in my previous lectures, for them I'm just telling at lower pH the amino acids tend to get protonated, and this affects the structure of the protein to some extent. So now what happens in this case is that both the proteins will be affected to some extent. Now in protein X, what happens is that it has mainly basic amino acids, because we can see from its high pi value, so it will be protonated. At lower pH, since the other residues in X will also get protonated, the net intermolecular repulsion due to similar charges will also increase as compared to your Y, and this will lead to your rapid unfolding of X. So therefore option C is correct, where you see that protein Y unfolds at a slower rate as compared to your protein X. So option C is right. Coming to the next set of questions, and I think this is the last set. So okay. So upon adding denaturants with a gradual increase in concentration to a protein solution, the absorbance of the protein increases as seen in UV spectrometer. This is due to why? What happens when denaturants are added? The change in concentration of the protein, the unfolding of protein, gradual exposure of tryptophan and tyrosine residues to the solvent, the increase in concentration of the denaturant. So this is an MSQ. So two options would be correct. Which are the two options? Oh, I'm telling you that also that two options are correct. Now which are the two options? C, that is the gradual exposure of tryptophan tyrosine residues to the solvent. Yes, that is true, because why is it true? Because what happens when denaturants are added to the protein? So yes, C is the correct option, but what makes these residues exposed to the solvent? So it is the when denaturants are added, the protein gets unfolded, because of which these tryptophan residues are exposed to the solvent and are out to the solvent environment. And because of these, the protein is the absorbance is has been seen to be increased in the UV spectrometer. So both the options are correct. Is B and C, the unfolding of protein and the gradual exposure of the residues to the solvent. The temperature at which the structure of protein breaks is generally referred to as melting temperature, boiling temperature, saturation temperature, and the Boyle's temperature. This is an easy one. Which is right? So when the protein is half natured and half denatured, what is that temperature called? So we call the temperature as the melting temperature or the TM. So therefore A is the right option. And coming to the last question, which of the following cannot denature a protein? Is it sodium dodecyl sulfate? Is it guanidinium hydrochloride? Is it iodoacetic acid? Or is it the urea solution? So which of these cannot denature a protein? See, yes. What does sodium dodecyl sulfate do? It gives a uniform negative charge to the protein, because of which the protein gets denatured and unfolds itself. So we have seen the use of SDS in SDS page when we run the protein and check the protein expression. Guanidinium hydrochloride is a reducing agent and it also hampers your hydrophobic interactions. We have seen urea performing the same role, but iodoacetic acid is an alkylating agent which cannot denature our protein. So yes, option C is the right answer. Okay, so that's all with the question and answer session. I thank you all for listening to my lecture and attending these live sessions. I hope this session would be beneficial to you people. Any other questions from your part? Okay, so that's all then. We can end the session over here. Thank you all for coming and listening to my lecture.